So my name is Zane, and my research was in analyzing protein sequence similarity networks. So I'll tell you what those are in a sec. Uniprot is a protein sequence database where researchers can upload any protein sequences they discover in their research. So what the team did before I came was they compared all the proteins to one another, and they built this table where we have protein 1, protein 2, and then the bit score is how similar they are. We only include them in this table if they're over a certain threshold. And then what I did is I took this table and I represented it as a graph or as a network where each node is one protein, and then the edges and the edge weights between those nodes are the bit score and how similar they are. So this graph actually had many different components. There's about 6 or 7 that were over 100,000 nodes, and there were probably about 1 million that were below 1,000 nodes. So we were looking at the bigger ones because there's more patterns that we could recognize, and there's less randomness. So the goal was to understand the structure of these similarity networks. So that means the connectivity properties, the structure of the similarity, so where are the very similar nodes and where are the less similar nodes within the structure, and then compare that to known network models. So we could see if there was any known network models that we could compare our data to. So examining the structure of the edge weights, so what we did was we built a graph called G alpha, where we made a threshold for what weights were going to be included in this graph, based on a proportion of the maximum weight in that component. So as we change alpha and we increase, you can see the number of components increases because we lose a lot of the connections, so the parts break apart. The average degree of nodes goes down because we lose a lot of the connections between the two nodes. And you can see these are two different components, but they have somewhat similar structures as we look at them here. So the next approach was analyzing more network properties. So the two important ones here are the degree distribution, which is just a frequency histogram of the counts of each degree, and then a k-core distribution, where a k-core is the largest subgraph of a graph, where every node has at least k edges coming off of it. So when we look at these histograms, we can see these again are two different components, but they have some similarities between the structures. So we can see the degree distribution sort of come down across like this, and then they sort of fall off at the very end. And in the k-core distribution, there's a lot of flat sections followed by very steep drop-offs. And even though they're not exactly the same, they still have those same features. So the third approach was looking at the k-core. We wanted to collapse the k-core into a single node, so we could see how this densely connected inner core of the graph interacts with all the nodes around it, all the edges outside. So what we did is we chose the first drop-off point where, so k equals about 100 here, and there's a drop-off. And we condensed that 100 core into a single node. And so here you can see before and after what happened. So after, it removes a lot of the drop-off at the end because a lot of the high-degree nodes have been removed, and it's a lot more linear without the huge drop-off at the end. So what are these results? Well, we found that they didn't fit any standard known network models initially. But after we removed the k-core, you can see this orange part is with the k-core removed. It's much more linear, and we can basically break it up into 3 different sections. And one of the things is it might even be technically 2 sections, but because we chose 100 core as the cutoff, we sort of have some residuals left in this graph that's caused by choosing 100 core, because it wasn't exactly a sharp cutoff. It was sort of rounded off at the top. So what does this suggest about the graph? Well, it means there's some sort of dense core in the middle, and then a less dense core around it, and then some nodes extending off of the outer core. And if we go look at these k-core distributions again, they suggest the same thing, because there's sort of a large port where a lot of the nodes are included, and then all at once, a large portion of the nodes get dropped off. So that suggests that there might be multiple layers of cores as we get down to the highest k-cores on the inside. So what's cool about this project is The initial research is in biology, but I don't understand a lot of the biology that they're using. But I was able to analyze these graphs, and hopefully my analysis and results will help the biologists understand something about these protein similarity networks.